Hello and welcome to today's GeneWiz Week webinar, Automated Microfluidic Platform for Circulating Tumor Cell Capture and Analysis. I am Damian Urena, Business Marketing Leader of Next Generation Sequencing Services at GeneWiz, and I'm very pleased to introduce today's presenter, Dr. Yixing Wang. Dr. Wang is the Chief Scientific Officer at Celsi Diagnostics and has extensive experience in molecular diagnostics with expertise focused on the development of cancer diagnostic technologies, including the first FDA-approved circulating tumor cell technology, CellSearch. Prior to joining Celsi Diagnostics, Dr. Wang was Vice President of Molecular Probes at Ventana Medical Systems, a member of the Roche Group, and Executive Director of R&D at Veridex, an oncology molecular and cellular diagnostic business of Johnson & Johnson. Dr. Wang has comprehensive experience on identifying molecular probes and developing and validating companion molecular assays. He earned his PhD in molecular biology from Cornell University. If you have any questions for Dr. Wang during the webinar, please enter them into the Q&A section and we will try to address as many as possible at the end of the session. Without further ado, please welcome Dr. Wang.
Thank you, Damon. Good morning, good afternoon, and good evening, depending where you are. I would like to share with you an automated microfluid platform for circulating tumor cell, or CTC, capturing and analysis. Let me start with a brief introduction about our technology platform. I'll then walk you through some applications we have developed to this point, including capabilities on CTC molecular characterization. Circulating tumor cells have been observed in cancer patients for a long time. They are actually tumor cells that are shed from solid tumors and get into circulation. Because they are rare cells and it's very difficult to capture them, CTCs have not been used in research and clinical studies until very recent years. How difficult is it to find CTCs? It turns out one tumor cell gets into circulation for every 10 million tumor cells. And this one tumor cell is then diluted among billions of the blood cells in circulation. As a result, the final concentration of CTCs is about one cell in about 100 million to a billion of the blood cells in blood circulation. Finding these CTCs is no different than finding a needle in a haystack. In the last several years, there are many technology companies that have been focusing on development of circulating tumor cell capturing and analysis platforms. From cytometry, fast scan imaging, immunomagnetic bead capturing, both for positive and for negative selections, size filtration, all the way to integrated microfluid devices. We believe that integrated microfluid device would provide some advantages over other technologies in the long run to provide a more sensitive and a more effective CTC platform for both research purposes as well as clinical applications. So how does the Celsi CTC technology works? Our core technology is built upon manufacture of a microfluid chip, which has the same dimension as a regular glass slide, meaning the results of the captured cell can be visualized under a fluorescence microscope. The principle of the CTC capturing is based on size and deformability of a cell. Our microfluid chip has more than 56,000 of individual trapping wells connected to the inlet and outlet channels. At the bottom of each well, there is a pore of 8 micron, allowing blood cell to flow through, but larger CTCs trapped in the trapping wells. In addition to size filtration, optimal fixation of the cells and the flow speed of the blood through the microfluid chip are also key to the technology, where we optimize these conditions. In a typical experiment, one can start with about 4 to 8 ml of whole blood samples. There's no requirement for pre-centrifugation nor lysis of red blood cells. The capturing step itself is also labeling free. However, when the cells are trapped on the microfluid chip, we do use various antibodies to identify the CTCs over the background leukocyte. Here's an example of CTC identification, in which we extended the same definition as the gold standard in the field. Pancytokeratin positive, nucleus or DABI staining positive, and CD45 staining negative. On the microfluid chip, we capture not only single CTCs, but also doublet, triplets, as well as cell clusters, as shown on this slide. The first experiment we did is to test the capturing efficiency and the reproducibility of capturing using the microfluid chip. In this experiment, we spec in tumor cells into normal blood samples from a normal donor, where we can see the capturing efficiency of these tumor cells is around 85%, with a CV of less than 7%. In this experiment, we tested multiple cancer cell lines, and the results are very consistent. The data has been published a couple of years ago. Next, we tested clinical blood samples from patients. This slide is a summary of CTC counts from samples of the patients with metastatic diseases, prostate, breast, and colorectal cancers. In total, there are about 128 samples from patients. Along the line, there are 200 control samples. Those are blood samples from normal donors. This result is also published in PLOS ONE last year. Now, one of the advantages of cell-C CTC system offers is to go beyond cell counts or enumeration and also provide capabilities on molecular characterization of CTCs. On this slide, the top panel shows some of the on-slide analysis, including the immunocytochemistry, DNA FISH, and mRNA FISH. These assays can be done on the captured cells read using the same microfluid chip. The bottom part shows some of the off-chip analysis, including PCR and sequencing. To do that, we have developed a protocol to not only capture the cell on the microfluid chip, but also retrieve the cells through a backflow procedure from the microfluid chip. In the next few slides, I'll give you some examples of these applications I just mentioned. This is a slide to show the immunocytochemistry, where we have tested a number of antibodies on CTCs after the cells are captured on the microfluid chip. These markers include biomarkers for breast cancer, prostate cancer, as well as EMT markers such as vimentin. Our goal here is to provide a menu of validated antibodies or reagents to the end user for their biomarker research. Similarly, we have tested on-chip DNA FISH and mRNA FISH assays with commercially available probes. On this slide, the left-hand panel is an image of HER2 DNA FISH assay on breast cancer cells using VISIS probes. The right-hand panel is an image of the mRNA FISH on prostate cancer cells using ACD probes. Both protocols are available by using the microfluid chip and the cell-C instrument. To enable off-slide molecular analysis such as PCR or sequencing, we have developed a cell retrieval procedure as illustrated on this slide. The experiment started with about 4 ml of blood. We first deplete background leukocyte by using the Rosetta Sep CD45 cocktail from stem cell. This would allow us to reduce the background leukocyte to about 100 to 200 cells. Then the PrEP is made on the cell-C microfluid chip to capture the cells. After the cell capturing, we use PBS to wash the microfluid chip and backflow the cell out of the chip into a test tube. At this point, the cells are ready for nucleic acid extraction and molecular analysis directly. The procedure is fairly simple and straightforward. The recovery rate is very high, and the number of the background leukocyte after the CD45 depletion is about 100 to 200 cells. Now I'd like to give you a couple examples. This is the data provided by one of our collaborators at Biotheranostics. In this study, our collaborator tested a gene expression signature using RT-PCR with breast cancer cells. The 92 gene signature, called cancer type ID, is a clinically proven cancer classifier to identify tissue origin for metastatic cancer. This test is usually done on tumor biopsy samples. The objective of this study was to determine the feasibility of crossover this tissue-based test to a blood-based test using CTCs. As a proof of principle, our collaborator spiked in SKBR breast cancer cells into whole blood from a normal donor and performed the cell capturing and retrieval using the microfluid slide. After the cell retrieval, the 92 gene signature was measured from the retrieved cells, and the data was analyzed using the algorithm they developed. The image on the left-hand side is just to show the antibody staining of the SKBR cells in green and the background leukocyte or white blood cells in red on the microfluid chip after the cell capturing. The most interesting result is the RT-PCR data by the heat map on the right hand. On this heat map, each column is a gene in the 92 gene signature, and each row is a sample. As you can see, there are two clusters. The top three samples are two blood samples as well as the spike-in cells, the starting material. They group together. On the second cluster, on the bottom, there's SKBR3 cancer cells, which is a positive control cells, as well as the cell retrieved from the microfluid chip. The fact that the 92 gene signature can be detected from the retrieved cell suggested the capturing and the retrieval procedure was successful. The next example is in fact a collaboration with GenWiz. In this study, we analyzed DNA samples from the retrieved cells by PCR-amplified sequencing. In this experiment, we tested two DNA mutations. One is a deletion in TP53 gene, and the other is a point mutation in androgen receptor gene, AR gene. We used two spike-in prostate cancer cell line, PC3 and LINCAP, to test these DNA mutations. Using spike-in samples, we were successfully in detecting both of the TP53 deletion as well as the point mutation of the AR gene. We're in the process of testing blood samples from metastatic prostate cancer patients at this time. These are important first steps to develop potential clinical applications using liquid biopsy assays. The data was recently presented at the AACR in April. As you may tell at this point, and I would also like to emphasize that these collaborations are through partnership with academic laboratories and industry partners. Here are some examples from academic labs in Johns Hopkins University, Thomas Jefferson University, University of Michigan, and Henry Ford Health Systems. We also have several industry partners, including Biotheranostics, GenWiz, STARTS, as well as international companies such as Capital Bio in China and Rajiv Gandhi Cancer Institute in India. We have also published the study on both analytical verification and testing of the clinical patient samples in peer-reviewed journals, as well as present these data at major cancer conferences. To sum up, here are some take-home messages. Although there are many technologies for CTC capture and analysis, we believe that integrated microfluid system offers a high degree and cost-effective automation. Our CTC system capturing efficiency is greater than 85% with a less than 7% variability. Besides cell counts and enumeration, our platform also allowed user to retrieve the captured tumor cells for PCR and sequencing analysis. I would like to stop here and answer any questions you may have. Thank you for your attention.
Thank you, Dr. Wang, for a very interesting presentation on circulating tumor cell capture and analysis. We will now take a few questions from the audience. Our first question is this technology available for early detection of cancer?
That's a great question. The whole promise of the CTC technology is to enable the patient care across the stream from early detection all the way to therapy selection and therapy choices. However, I would say the research application as well as the clinical application today for CTC has been limited to metastatic tumors. There are a couple of reasons, both scientifically and clinically. Scientifically, I think it's hard to pinpoint at what time or what stage of tumor progression the CTCs shed from tumors into the circulation. And they probably vary from one cancer type to another. Clinically, I think the limitation has been the technology. Most of the technology, including the one I mentioned for CTC capture analysis, do not offer the sensitivity for early detection of the cancer. Even in metastatic disease setting, these capturing efficiency is around 70, 80, and perhaps lower 90% at the time. So that's just highlight the requirement for further develop such technologies into research settings as well as clinical applications.
Okay, thank you, Dr. Wang. We have a second question. What other technologies are available to capture CTCs and what are their advantages?
Yeah, as I mentioned, there are a few approaches that has been used to capture and analyze CTCs, including size filtration, as I talked about today, as well as magnetic beads coupled with antibodies. One of the systems I want to mention is Cell Search, the first and only FDA-cleared CTC system for CTC analysis in a clinical application. And some of the other technology require fast imaging, basically start from a blood smear, then using a fast imaging scanner as well as a complex computer algorithm to identify the CTC in the large background of the blood cells. Each of the technology have advantages and disadvantages. I think most of these technology are still at the research or early stage of research applications. I think there's a long way to develop those as routine cancer diagnostic tests in the clinic.
Thank you again, Dr. Wang. Our third question is, can viable cells be captured with this technology for in vitro culturing?
The answer is yes, and yes. There are procedures for capturing not only fixed cells, but also live cells and viable cells for different research applications as well as clinical applications. As I mentioned, some of the imaging-related analysis, such as immunocytochemistry, FISH assays, those are required fixative to treat the cells before these cells can be used for antibody staining or probe hybridization. So they're best applications for the fixed cells. On the other hand, for most of the molecular analysis, as well as in vitro culturing of circulating tumor cells, it would require live cells, and that can be captured as well as retrieved from these technology platforms. And perhaps one of the differences is the sample collection, the blood collection tubes. For fixed cells, one can use some of these blood collection tubes that contain fixative, which enable the sample to be processed within several days. However, for molecular analysis as well as cell culturing, one probably have to use EDTA tube, which need to be processed within 24 hours.
And we have time for one more question. What cancer types could be screened through CTC capture?
That's a great question. I think in the last few years, many of these solid tumor has been tested for detection of circulating tumor cells. If you look at the 4 major solid tumors, breast cancer, prostate cancer, colorectal cancer, and lung cancer, which counts for more than 50% of the newly diagnosed cancer cases in the United States, perhaps similar data in other regions and countries. Many of these solid tumors produce CTCs. The frequency of the CTC detection vary from cancer to cancer. For example, some of the major solid tumor like breast and prostate cancer produce more CTCs at higher frequency, mainly in metastasized bone tumors. And other cancer types such as colorectal cancer may have a lower frequency of CTC detection, but they do have CTC identified. So I would say majority of the solid tumor will have CTCs.
Thanks again, Dr. Wang, for your insights. This is all the time we have for today. Thank you to everyone in the audience for attending this webinar. We will be addressing any remaining questions offline. Please visit genewizweek.com for more webinars and other activities.